Well, thank you very much, Ed, for the kind introduction. And I would also like to thank Josik and Curie for the kind invitation. It's an honor and pleasure for me to give here a talk, and it's also an honor to be part of this ICAN program, especially taking into account that I'm coordinating a sister program in Germany. So it's also dedicated to establish a link between neurotechnologies, neuroengineering, and physiology in principle, or experimental neuroscience. Well, when I was asked to give a talk, I decided to choose a very general title by telling you something about how neural networks develop in the brain under physiological but also pathophysiological conditions. And then I decided, also taking into account the recent development in my group, the recent data, to be more focused and to talk today about the cellular substrate of these brain rhythms during early development. And to talk after Howard and Curie, it's certainly a big burden, but it's also a big chance because I can save time in my talk. So I can skip entirely my introduction, more or less. So I will not tell you once again how important the communication between brain area is, how important brain rhythms are, how they are a neural code for allowing the representation of different memory items. You heard about this, and you heard really from the experts. But what I would like to talk today a little bit more, or to point out, is that everything what we know about this coupling, rhythms, communication, we know for the adult brain. But there is one big question: what about the developing brain, or how the brain reach these impressive cognitive abilities? How long is a way to get there? And what's going on this way? Or to put it more precisely in a question, how oscillatory activity, coordinated spiking patterns contributes to the development of cognitive abilities by shaping neural networks. This is a quite, apparently a quite simple question, and I also thought like this when I started my postdoc and trying to address this question, and later on with my own group. The prerequisite for that is, of course, to know whether a developing and immature brain has oscillatory patterns or coordinated patterns of activity. And apparently it has. In all species that have been investigated, there are these oscillatory patterns. They look totally different from what we know from the adult brain. We don't have continuous activity. You know, if you record in a brain, on the surface of the brain, what you get is always the rhythm that switches between different frequency depending what the brain or what the person or the animal is doing. However, during development, the situation looks totally different. The activity is not continuous but intermittent. There is a flat line alternating from time to time with such periods of oscillatory discharge. They look like a spindle. This is why they have been called spindle bursts in rodents, and they are highly reminiscent to the human delta brushes that have been investigated since many, many decades in premature children. And you can see, looking at these discharges, that they look very similar. And this is true for also other species that have been investigated. So for the rest of my talk, I will focus on these patterns of oscillatory activity as well as on the spiking patterns that are correlated with that or associated with that. And everything that I will present to you are data recorded under in vivo conditions in head-fixed or freely moving animals. And they have been, and for that, when I mentioned that, I thought it's a simple question to address this one. I did not consider the fact that everything that you know for doing in vivo electrophysiology in an adult mouse or rat, you must forget about it when you start to work with such tiny animals. So everything needs to be miniaturized, so more or less you must develop your own techniques for doing that. We were quite good in solving the technical problems, at least for most of the questions that we addressed. And we ended up, or meanwhile, we have a quite clear picture how at system level, at the neural network level, the interactions between different brain areas are established early in life, how they look at the neonatal age. When I'm talking about neonatal age, I'm talking about the first postnatal week in mice and rats. So everything that you will see today corresponds to this period. Well, what we know, and I will try to make a kind of very long story short, most of the data have been already published. What we know is when we focused on a network that is dedicated to cognitive processing, and you heard about, including the prefrontal cortex and hippocampus, what we know now is that there are already at this very early stage of development complex interactions between these areas. We could show, so if you look at the activity patterns, you see that this is for the prefrontal cortex. We focus on the prelimbic subdivision of the prefrontal cortex, and this is the activity that corresponds to the hippocampus. Again, there is nothing going on from time to time. These discharges, they range in different frequency bands. They have a typical ground rhythm in theta range, so 4 to 10 hertz, and they have superimposed on that faster activity in low beta, low gamma beta range, so around 20-30 hertz. And there is a lot of spiking associated with this oscillatory discharges. At the hippocampal side, the activity is again discontinuous. What you have at this age is a typical theta discharge. We call them theta bursts. From time to time, there are also sharp waves superimposed on the theta rhythms. Well, we could show that the hippocampal theta burst acts as a kind of driving force for the local entrainment of the prelimbic cortex. And this communication we use for those different methods of calculating the synchrony, causality, and so on. But interesting is that this drive is unilateral. There is no drive back, so there is no information back directly from the prelimbic cortex to the hippocampus. And we were quite surprised about this because we thought, well, prefrontal cortex must communicate back. It's very unusual in the brain to have such a unidirectional coupling. And we started more recently to search for areas that can act as a kind of relay from the prefrontal cortex back to the hippocampus. And we searched quite a lot, and we finally focused on two areas. The first one was the lateral entorhinal cortex, and the second one was the nuclei of the midline thalamus, especially nucleus reunions. And we could show that the entorhinal cortex is not a relay station. It's it's driving both areas. It's driving very strongly the hippocampus at this age. And it drives to also a certain amount the prefrontal cortex. But the areas that act as a relay, it's a midline thalamus, the nucleus reunions, so the information from the prefrontal cortex goes back to the hippocampus. And the data were recently published on this. So, summarizing, we can say that the hippocampus drives the oscillatory entrainment, this local entrainment of the prefrontal cortex. Like you can imagine, it's a master that times the organization of the prefrontal cortex, and the thalamic nuclei acts as a relay station at this age. Everything that I showed to you was obtained at a network level. So I mentioned, so we use measures for assessing this interaction. We use synchrony, we use causality measures. However, what I think it's very important, there are two aspects. The first one is this coupling relevant for behavioral abilities? And the second question is, what is a substrate of this coupling? Well, for the first question, I can give you only very few data, and we are working for the moment exactly to answer this question. How relevant is early activity patterns or the early activity patterns in the developing brain for the cognitive performance at later stages of development or adulthood? We have some preliminary data that when we interrupt the coupling between or the drive from the hippocampus to the prefrontal cortex in a specific period of life, that the animals are not able or have a poorer performance in a cognitive test that is one of the first cognitive tests that a rat can perform, the recognition memory. And it's very good for rats and mice at early juvenile stage because you don't need to train the animals for doing that. So they love to recognize the objects, so you don't need to train them. You just put them in a behavioral testing apparatus, and you can test them also at 2 weeks, 3 weeks of age, at a period that corresponds to the juvenile stage. So the first question is somehow unanswered, but at least I can offer you some answers for the second one. What is a cellular substrate? Which are the neurons that contribute to this prefrontal hippocampal coupling? And how this network entrainment looks like? So we started for this, to answer this question, we started firstly with what I call correlative approach. We performed in this neonatal animals simultaneously local field potential recordings using the previously introduced multi-site recording electrodes and all possible configurations designed together with NeuroNexus or with a standard configuration that we could get from NeuroNexus. And we did this simultaneously with patch cam recordings in vivo from these neonatal animals. The animals that we recorded were in a sleeping-like state, meaning that so everything that is shown here is not when they freely move. I must tell you, during the first week, they don't like to move so much. So they sleep 80% of the time. And if I put a neonatal rat or mouse on this table, I will finish my talk and will still be on this table. Probably this is not happening with an adult one. So we use this as a paradigm, so recording paradigm, and you can see we stain these cells, so we fill them with a marker so we can identify them where they are in the brain, and also we can classify their neurochemical profile, so whether they were GABA positive, for example, or GABA negative. And this is a trace of they were located in the proximity of the field recording electrode. This is a red trace that you can see here and here. This is a GABA negative neurons, has a typical pyramidal shape, and the other one was a GABA positive neuron, that it was an interneuron. And then what we did was to correlate the local field or the temporal pattern in the local field and for the intracellular synaptic inputs of pyramidal neurons and interneurons, as well as the local field with the spiking patterns of these neurons. And we correlated this, we calculated these funny methods that will not go into details. So synaptic current triggered LFP average is nothing else than aligning all these synaptic events to a specific phase of the oscillatory activity. So by doing all this analysis, it allows us to determine temporal relation between a single neuron and the field, and by this correlation, we could, and we end up with a diagrams that allows us to say which are the interactions or which interactions contribute to a specific pattern of activity. As I said, it's just a pure correlation. So we don't have a clear proof that this particular neuron is involved in the generation of theta or beta low gamma activity, but correlative, we could show that they do this. The theta activity results from the long-range coupling between pyramidal neurons. We have no idea where this pyramidal neuron is sitting, but it's not sitting in the prelimbic cortex. We hypothesize that it is in the hippocampus. But as I said, we have no proof for that. And also some local interactions between interneurons are important for that. A different picture is true for beta low gamma activity. In this case, there is necessary to have a glutamatergic drive on prelimbic interneurons. And as you can see, this is a nice effect of this method, of these patch clamp recordings. One can also say which kind of receptors, if you have the synaptic currents, you can say, okay, what kind of receptors are involved, whether they are AMPA-mediated or NMDA-mediated synaptic currents. So this is somehow already an advancement. So we know a little bit how the network looks like, that it's involved in the generation of different activity patterns. However, this was a correlative approach, and optogenetics is now the method of choice for each kind of project. So we thought, well, it's a good method to use it also for elucidation of the circuitry. However, we face two major problems. The first one is we need suitable light-sensitive proteins that are so they can be used for these neonatal neurons to activate them, or even better, to silence them. And the second problem was how to target this early neural subpopulation during development. Well, to have light-sensitive proteins, we wanted to have a light-sensitive protein that follows also at faster frequency, because we don't want to look only at the theta range, but we wanted to look at the beta low gamma range. So the chanrodopsin, the classical chanrodopsin, are not so well suited. So together with our collaborators, Peter Hegemann in Berlin and Thomas Oerdner in Hamburg, we used for the activation of neurons a double mutant of the chanrodopsin. It's a so-called ATTC mutant. It has a big advantage that follows very good high frequency of stimulation. So it has a very good kinetics, it's fast, and it's very efficient, so induced with high precision action potentials. For silencing, we're starting now to work with a recently developed eye clock. However, as the preliminary data shows, and as we expected, a chloride channel is certainly not the best choice for developing neurons when taking into account that GABA is depolarizing at this stage of development. So we are now trying to use a prototype for potassium channel light-sensitive proteins for inhibition. Well, what about the methods for selective targeting? Well, everybody doing optogenetics knows simple viral vectors. Well, it's not so simple because we need to have good and stable expression already immediately after birth or few days after birth. Most of the viral vectors need quite long time. I think the fastest is something between 5 and 6 days, already too late for investigation of neonatal period. So we thought, well, viral vectors would be nice. It's a little bit, it's much easier than what we were forced to do. But on the other hand, the methods that we choose seems to be, once established, very reliable and ensures not only cell specificity, but layer and area specificity. And I'm talking about in utero electroporation. So we build these vectors, including the channelrhodopsin ATTC, and then applied specific protocols at specific embryonic stages for targeting layer by layer the prelimbic cortex, layer 2-3 or layer 5-6. Or we could target also the CA1 area using, as I said, a different protocol, the paddle, the position of the paddle being slightly different, as you can see on these diagrams. And the results were quite surprising. So I never thought that they are so well targeted. So you can see this is for the layer 2-3. It's a double staining, NOI-N for neurons, and the T-dimer is a fluorescent marker in our vectors. And you can see that they are always co-stained. So all the transfected cells are neurons. And none of these transfected cells was positive for GABA, so they are all pyramidal neurons. So it's really, we were able by these methods to specifically transfect the pyramidal neurons in the layer 2-3. For the layer 5-6, this transfection was, also the level of transfection was relatively high. However, not only due to the migration pattern of neurons, of course, not only layer 5-6, but also some neurons were transfected in layer 2-3. And this is a summary, and this is for the hippocampus. These are data that we are acquiring for the moment, so I don't have here the quantification for the CA1 for the moment, but again, it is very clear that CA1 pyramidal neurons are transfected. And the level of transfection was surprisingly high, so we are dealing with 35% of the pyramidal neurons were transfected, and this is also makes very reliable then the activation of these neurons. Well, what's going on when we activate them? Before we did this, we performed several control experiments. This was necessary because, as I said, this is neonatal neurons. We wanted to test, first of all, what's going on if we transfect in these neurons a light-sensitive proteins. Maybe this changed the entire properties, the spiking or the membrane properties, active, passive, whatever. So we tested this, and apparently there is no difference. So the neurons behave absolutely normal. They have normal membrane properties. They spike normally. They have the same spiking pattern as the non-transfected neurons. On the other hand, we established how to stimulate these neurons in a reliable way, how long should be the pulse, how intense the light, and so on. So all these parameters that for the adult have been established need to be established also for these very young neurons. And then what we did was to insert, this is how it looks like. So we have the multi-site optoelectrode, and at a certain intensity, we can get reliable spiking activity. And we tested, also using a protocol that has been developed in the lab of Joshua Gordon, that none of these stimulations, the intensity, never lead to a heating effect that could also induce spiking in these neurons. So we were, with a heating effect, we were below the levels that would induce spiking activity in these neurons. So after performing all these controls, we thought, let's see what's going on when we stimulate the neurons layer by layer, and also the pyramidal neurons. So everything that I showed, it's for the pyramidal neurons. So when we look at the layer 2-3, ramp stimulation, light stimulation in ramp form leads to an increase of the spike, and that also persisted after the ramp. So it's a short time plasticity that was induced. And when we look now at the, this is multi-unit activity, but the same is true also for the single one, normalized counting, we ended up with this picture. So you can see that during the stimulation, it's an increased firing that persisted also to a certain extent after stimulation, post-stimulation. But what's very interesting was the fact that we have here a clear peak at around 16 Hertz. It's exactly the dominant frequency that the beta frequency range has in the oscillatory activity. And this is and this spiking pattern is not intrinsic. So it's a network effect. We tested this. We need sinus waves. So it's not the intrinsic pattern of the layer 2-3 neurons. This type of spiking was absent when we stimulated by ramp the layer 5-6. In this case, there is just a broad increase in the spiking rate, also persisting post-stimulus. And this was not the case when we stimulated CA1 area. In this case, it was again an increase in the spiking of pyramidal neurons in this area, but it was broad over the entire beta range, so from 4 to 12 hertz. It's quite broad and has a tiny peak around 25 hertz. So this data already suggested that apparently layer 2-3 neurons, confirming our patch clamp data, are involved in the generation of theta activity. Are involved in the generation of beta activity, at 16 hertz. And to test this, what we did was to look at the field potential during RAM stimulation. And indeed, what we observed is that during RAM stimulation, it was a significant increase in the power of beta range. And after the stimulus, it was like a general excitation of the network. And the increase was present in all 3 frequency bands, theta, beta, gamma. The picture, of course, what is also interesting is that the phase locking in the beta frequency range was significantly increased after stimulation. This is not true for a layer 5-6. In this case, as I said, it's similar to what I presented to you before for the spiking data. There is an overall increase in all frequency bands, so it's more excitation, more activity. And for and again, there is no difference in the coupling strengths between units and the phase of oscillatory activity in none of the frequency bands. And the last one in the hippocampus, again confirming the patch data, what we observe is that after stimulation there is a slight increase, but this is general in all frequency bands and confirms the fact that stimulation of pyramidal neurons in CA1 area are not the main driver for one of these activity patterns. So again, here there is no difference in the coupling strengths. So if I summarize what I presented until now, I would like to say that the hippocampal theta bursts are the drive for the prefrontal cortex. And now if we look at how the cellular substrate for these 2 types of activity are the theta oscillation, beta oscillation. Theta results from this long-range coupling of pyramidal neurons, to a certain amount also from local interactions between interneurons, but we could not confirm this by optogenetics. We need to address also interneurons, but it seems to be a little bit more difficult at this age, having something to do with their migration patterns and how easy it is to address this by neutral electroporation. And the beta log gamma oscillations require the activation of layer 2-3 neurons. This is what both the patch clamp data, the correlative methods and the causal methods, optogenetics revealed. Well, at the beginning of my talk, I said I would like to present you something about the neonatal rhythms in health. This is what I did until now. But I would like to say a few words in the remaining time about the disease case. And to be honest, a matter of fact, the reason why we performed all this part of the study was to have the controls for the questions that now it comes, because we wanted to understand what's going wrong in several neuropsychiatric disorders that have been reported to be neurodevelopmental. And probably the most typical example is schizophrenia. Schizophrenia is known to be a mental disorder for which the etiology is established very early. So there are 2 factors that come together or should come together, defect genetic background and stressors during development. And it's considered that the abnormality, so the brain is abnormally wired because it has a developmental impairment. It's something during development goes wrong. And the questions that we wanted to understand what exactly goes wrong during development. So to address this question, we faced, firstly, it was the good news was the fact that in humans, it has been already shown that there is an abnormal coupling between prefrontal cortex and hippocampus. A group of Meyer Lindenberg in Mannheim gave important proofs for that, experimental proof in several groundbreaking papers. So there is indeed an abnormality within this network. These were adults, these were patients, so already with a disease, with a first psychotic episode. But the bad news is that schizophrenia is probably the most human disorders that exist. And the question that arises is how to establish a link between a mouse and a schizophrenia patient. And to be honest, we cannot. I mean, I remember Akira Zawa gave a talk on this topic and said something very funny. I listened very long to this schizophrenia mice, but they never told me that they have hallucinations. So with other words, every schizophrenia model, it's a bad model. However, each of these models can contribute or can help us to address a question that we cannot address in human patients, because we cannot invasively look at which neurons are impaired in a human brain. So there are some possibilities to link the two species by common etiology, so to mimic a genetic background, abnormal genetic background, and environmental stressors during development. One can do this. We can look at common symptoms, of course, no hallucinations, but a cognitive impairment, for example, working memory deficits, can be also observed in a mouse or in a rat. And third, common dysfunction. And this is one of the first studies that have been done on this field. It is also from the group of Joshua Gordon together with Joseph Gogos, that showed that in a mouse model of schizophrenia, there is a similar abnormal coupling between the prefrontal cortex and the hippocampus. And these were adult mice. So indeed, it seems that the network, this coupling between the two areas is disturbed. So what we did was to investigate mice models of schizophrenia, different mice models, but we did this not at adult stage, but very early. And we look at the genetic mouse model, so only abnormal genetic background. Doesn't matter. This example was for disrupted in schizophrenia 1 gene. We had also several other mice models, genetic ones. We used a so-called environmental model, where an environmental stressor was the factor causing the disease. We use a maternal immune activation by mimicking a viral infection in the mother during pregnancy, during gestation, and then look at the pups. And we combine these two factors in a so-called dual hit. And I summarize this, the whole work, what we did, and we ended up with a conclusion that only when these two factors come together, indeed, there is a massive change in the network properties, in the activity patterns, both at the level of the prelimbic cortex and in the hippocampus. But what was even more interesting is that the coupling between these two areas is disturbed. The drive from the hippocampus to the prefrontal cortex is significantly decreased when these two factors come together. Now the question is why it significantly decreased, and we started to address this, and the preliminary data that I want to share with you are coming from this optogenetic manipulation. We did the same experiment what I showed you before for the normal mice, so no schizophrenia model mice, let's say, the controls, and you remember this peak after stimulation with the ramp when the layer 2-3 neurons were activated by light. Now we did the same experiment in these dual hit animals, and to our surprise, there is no peak at 16 Hz. There is an increase in firing of these neurons, but there is no specific activation in a frequency band. And this fits very nicely with the data that I showed before, that is a significant change in the power within this frequency range, is the amplitude and occurrence that was much more prominent than for the theta range. And certainly now it's a lot of work to be done, and we have already on our to-do list what exactly, or what is the consequence at the behavioral level when this optogenetic manipulation or these changes are done during early stage of development, how the behavior is affected at adulthood. Well, by saying this, I would like to finish my talk thanking for the generous support of several funding organizations that supported our work over the years, especially the German Research Foundation, more recently also the ERC. And I'd like to mention that all the work that I presented today has done by 3 very talented PhD students, Sebastian Bitzenhofer and Joachim Albeck, established the entire optogenetics in neonatal animals in my lab. Joachim is here at the meeting and has a poster on this issue. Kai Sieben developed many of the analytical routines that we used. Again, a totally new chapter. Nothing that is known to work for the adult can be applied in analytical terms, can be applied for this neonatal patterns of activity. So it was necessary to develop all the routines and analysis. And Henrike Hartung and Christoph Lindemann are the 2 postdoctoral fellows that work on the schizophrenia project. This is my group. Officially, we are the Development and Neurophysiology Lab. A non-official name is a nested gamma spindle burst. It's a typical pattern of activity that is present only in the prelimbic cortex during development. Okay, thank you very much for your attention.
There are 30, 40, 50 places in the world that can do this. I'm not sure there's anyone else that will do it. And so IMAX says, well, we're going to make switched and active probes, and we'll give you 180 channels.